Hello everyone, my name is Tulani Heva Vitana and I'm a PhD student at the University of Saskatchewan's Department of Computer Science. Today, I'm thrilled to walk you through our research on the transcriptome analysis of Brassica polyploids, specifically focusing on how allopolyploidization affects gene expression. Why are we interested in the Brassicaceae family? These plants are not just crucial to agriculture, providing us with vegetables, oils, and biofuels, but they are also fascinating from a genetic standpoint. The genus Brassica includes species like Brassica rapa, Brassica nigra, and Brassica oleracea, which are known for undergoing natural hybridization and polyploidization. This leads to the formation of new species like Brassica napus, Brassica juncea, and Brassica carinata, which are critical for enhancing agricultural productivity. These species have undergone a significant evolutionary event known as Brassicaceae-specific whole genome triplication, where their genomes didn't just duplicate, they tripled. This resulted in 3 distinct subgenomes within the same nucleus named least fractionated, moderately fractionated, and most fractionated. These subgenomes contribute to the genetic diversity we observe today, which is pivotal for the adaptability and resilience of Brassica species. What exactly are subgenomes? They are like different chapters in the genetic book of a polyploid organism. Each chapter originates from different ancestral genomes and can influence the overall storyline of the organism's development and function. In the case of polyploids, genes from one subgenome may be expressed more than those from others, leading to what we term subgenome dominance. Our research focuses on comparing the transcriptomes of Brassica tetraploids and their diploid progenitors. We are particularly interested in how gene expression varies between different tissues, subgenome types, between diploid progenitors and their tetraploid species. The key objectives of our study are to assess how gene expression levels vary across different tissues influenced by the BRWGT event, to understand how gene expression patterns differ based on the parental genomes and tissue types, to explore the extent of genome rearrangement and gene loss and how these factors influence subgenome dominance. We asked several research questions. How does gene expression in allotetraploid genomes differ compared to that in diploid genomes? Are there differences in gene expression among different tissues in subgenomes? How do genome rearrangement and gene loss impact subgenome dominance? Our methodology began with the alignment of quality filtered row reads from pollen and stigma tissue transcriptomes of used triangle Brassica species as shown in figure 2. We utilized DagGener for identifying synteny genes and SyntanyLink tool to categorize these genes into their respective subgenomes. Our analysis investigated expression dominance by measuring and comparing the overall expression levels of corresponding homologous gene pairs in allopolyploids against their original expression levels in diploid progenitors. This required us to calculate mid-parent expression values, which represent the average expression levels of each gene pair from the diploid parents. Additionally, we assessed homologous expression bias, where we considered the relative expression of each homolog to their diploid progenitors to identify discrepancies in expression levels between gene copies inherited from the diploid parent. We examined the extent of genome fractionation and rearrangements using metrics like fractionation rate and DCJ distances, which were calculated from Unimog software. In our results, we found significant differences in gene expression between different tissues like stigma versus pollen, with stigma tissues showing higher gene expression levels, suggesting tissue-specific regulatory mechanisms. The least fractionated subgenome showed dominance across all species and tissues, as shown in figure 5. Interestingly, figure 2 shows that the subgenome inherited from the maternal parent showed expression dominance in all tetraploids and had the lowest fractionation rates, highlighting a potential role of maternal factors in subgenome dominance in tetraploids. Literature shows some variability when it comes to maternal lineage. For instance, in the case of Brassica napus, some studies identify Brassica oleracea as the maternal parent, while others suggest it's Brassica repa. However, when we look at Brassica juncea, it's a bit more straightforward. Brassica nigra is consistently recognized as the maternal parent. The same goes for Brassica carinata, where Brassica nigra also serves as the maternal parent. This mix of variability and consistency really underscores the complex role that maternal factors play in shaping subgenome dominance and gene expression patterns across these species. Homologous expression bias varied between tissues, indicating complex interactions between environmental and genetic factors in determining subgenome bias, as you can see in Figure 3. Our analysis of the DCJ distance matrix showed that the subgenome with higher genomic rearrangements demonstrated subgenome dominance, as shown in Table 1. In conclusion, our study provides substantial insights into how complex genomic rearrangements in Brassica species influence their adaptability and functionality. The variations in gene expression between tissues and types of polyploids underscore the dynamic and intricate nature of plant genomes. Thank you very much for your attention.